Okay, so before we talk about how to do a spore stain, I want to talk a little bit about what a spore or an endospore is. And some of you may have gone into some depth about that in lecture, so this will be review, some of you not. All right, so first of all, let me remind you what spores are for bacteria. Spores, or you could say sporulation, is a survival mechanism. Okay, what it not is, that makes sense. What it not is, it is not a reproductive strategy. Okay, so sporulation in bacteria is quite a different process than sporulation in fungi. Fungi sporulate to reproduce. You get hundreds of spores released from, one vegetative structure, we might say. With bacteria, as you'll see, we get one spore from one metabolically active cell. All right, so that's not considered reproduction. Okay. So we'll start out here. I'm going to draw a rod-shaped vegetative cell because, as far as I know, all of the bacteria that do form spores are rod-shaped. There may be some exception there. So here's a vegetative cell. Remember what I mean by that. Vegetative just means it's a metabolically active cell. Okay, and if that bacteria cell encounters some kind of harsh conditions, talk about some examples of that in a minute, it will undergo sporulation. So that means it's going to begin the formation of a spore somewhere within that vegetative cell. And sometimes we call that stage the endospore. Okay, so we won't go through, all the genes that have to be turned on and off for that to happen, but it's a pretty complex process. What kind of harsh conditions? Hopefully you can think of some harsh conditions, any condition that wouldn't be ideal for a bacterial cell. So what are ideal conditions for a bacterial cell? Well, it's different depending on what cell it is. But usually warm, moist conditions are going to be ideal places for bacteria to reproduce and survive. So anything that's not warm and moist. Okay, so some examples of harsh conditions might be dry conditions. Bacteria don't like to dry out. Low nutrients availability. Okay, that can certainly pose a survival risk. High temps, usually not low temps. It's not going to kill a cell to be cold. More often high temps, maybe high or low pH fluctuations, radiation exposure, like ultraviolet light exposure. Maybe exposure to some kind of chemical, disinfectant. I'm sure you could probably, the list could go on and on, but there's just some examples of harsh conditions. Eventually, that vegetative cell will fall off, and you will be left with a free spore. Now, sometimes we're moving more and more towards calling this an endospore as well. Just to differentiate a fungal spore and a bacterial spore. Oftentimes we just call a bacterial spore, whether it's free or in, still within a vegetative cell, we just call them endospores. Okay, and an endospore can stay as an endospore basically indefinitely. All right, the spore coat here is really tough. Lots and lots of layers of protection around the DNA, which would be inside here. How long can that endospore persist and still possibly be viable again? We don't even know. Certainly years, certainly decades, centuries, even thousands of years. We've found spores that we believe to be thousands of years old on mummies, and they've still been able to germinate. So, a long time. Say conditions eventually improve. Okay, so nutrients become available, for example, then that spore or the endospore would germinate back into a vegetative cell and continue on being metabolically active and reproducing into more vegetative cells, that sort of thing. Okay, keep in mind this spore cycle takes somewhere between 6 and 7 hours. There's a little variation there. So, if you throw some vegetative cells into a boiling water, right, they're going to die, all right, because they can't sporulate fast enough to withstand, certain conditions. We think that a lot of cells just kind of, whether they encounter harsh conditions or not, they just sort of regularly go through this spore cycle. So that if environmental conditions do change rapidly, even though most of the vegetative cells would die, if there happened to be a few spores in that cell population, those would withstand the harsh condition and then be able to germinate when those conditions improve. Okay, just noticed. Let's say spores are a survival mechanism. That sounds like better English. Let me just fix that there. There, all right, I'm a little more satisfied there. Okay, so what kinds of cells are able to sporulate? There's two of the most famous genera for being able to sporulate are Bacillus and Clostridium. Sporulation is not common in the bacterial world. We said lots and lots of cells form capsules. Again, there's really only one genera that's acid-fast, and there's really only a few that are able to form spores. And some of the other genera that you might read about may have originally been considered Bacillus or Clostridium, and then we've kind of split those genera up a little bit. Okay, so just some examples here. Bacillus Subtilis, that's a species that we typically use a lot in lab. It's a cell that's it's a species that's found in the soil. Most of the bacillus are going to be environmentals. Okay, they're soil organisms. Both Bacillus and Clostridium are gram-positive rods. Okay, so most of the rod-shaped organisms we've worked with in lab have been gram-negative, like E. coli and Klebsiella and Proteus and all those, but there are gram-positive rods as well. They're usually a little bit larger than some of those enteric organisms. Sorganisms. Okay. All right, so Bacillus, most of the Bacillus, like I said, these are aerobes for the most part, or all of them as far as I know. They're mostly harmless. The big exception is Bacillus anthracis. Okay, that's the causative agent, that's the CIS, that's the causative agent of anthrax. Okay, might have heard of that. Clostridium, on the other hand, there's a lot of really nasty pathogenic Clostridium. These are primarily anaerobes. In terms of what cells are we going to be staining in class, some semesters we stain Bacillus subtilis, some semesters we use Clostridium Sporogenes. I don't know at this point which one we'll use. We always used to use Bacillus subtilis, and then we started having trouble getting that organism to actually sporulate. So it wasn't terribly exciting when we do our stains. So more recently, we've been doing this Clostridium Sporogenes. It's one of the Clostridium that are non-pathogenic. Some other interesting ones, Clostridium tetani. That's the causative agent of tetanus, sometimes called lockjaw, or Clostridium botulinum. I think there's another I in there, not sure. Botulinum or botulinum. That's a causative agent of botulism. Okay, both really nasty infections because the Clostridium produce a very potent neurotoxin. So for tetanus, that neurotoxin keeps our muscles from relaxing, makes a very rigid type of paralysis. And botulism is a form of food poisoning that toxin produced keeps our muscles from contracting. So it's, again, kind of called a flaccid paralysis. But eventually patients aren't able to flex their diaphragm in order to breathe. So both really nasty infections. Clostridium Difficile is another one, causes the disease known as C. diff, a really nasty enterocolitis, diarrhea. Really pretty nasty and can be pretty chronic. Okay, so those are just to give you an idea of some of the kind of organisms that form spores. How are we actually going to do a spore stain? Well, you're going to be staining two different organisms, so we're going to have two different slides to spore stain, and you're going to want to label them because you got more than one slide eventually here, but you're going to start out with a drop of water. Again, the smaller the drop of water you can get is usually preferred. And on one slide, you're going to mix in, say we're using Clostridium Sporogenes, whatever our example of a spore former we use. And I would say you can use your needle to mix that in there, just because when you use your loop, you just tend to get way too many cells, and they're all pressed together, and you can't see their shape. Okay, and with your other slide, we're going to use an example of a non-spore former, Staph epidermidis. All right, so again, you might want to label those, CS or SE or whatever, because you're going to have several slides to work with today. Okay, make sure you heat fix those cells once you get them mixed into that drop of water, because we want them to adhere to the slide. Okay, then when you're ready to start your staining, the first stain that you're going to use is Malachite Green. You're going to flood your smear with Malachite Green. And there's really nothing special about Malachite Green. It's a basic dye. What's special about the Schaefer-Fulton spore stain procedure is that we are going to apply heat to the cells with the Malachite Green on them in order to really hammer that stain into the spore coat. Okay, so the heat is necessary, again, to break down the spore coat a little bit. Enough so that we can get the stain to adhere to it. All right, so remember, we are not causing the spores to form by applying this heat or heat fixation. Remember, it takes 6, 7, 8 hours for the spores to form. So just by heat fixing the cells or applying heat for 5 minutes, that is not causing the spores to form. We are hoping there are spores already in the mix of cells that we're using. Okay, and I'll show you how to do this in class, but we're going to put our slides over a little tray of steaming water. Okay, so that steam hopefully will break down the spore coat enough to get the Malachite Green to stick to it. Okay, after about 5 minutes, you're just going to give those cells a good rinse. It's not any other type of decolorizer necessary here. And the last step, it really is just adding a second dye, safranin, in order to dye the vegetative cells. For whatever reason, the vegetative cells don't take up that malachite green heat combination very well, so the spores should stain green, and safranin, we leave that on somewhere between 1 and 2 minutes. Hopefully that should stain the vegetative cells. Give that a good rinse, and of course we're going to blot it dry before we put it on your microscope stage. So think about what we hope to see here. Okay, if we think about what our spore formers should look like here, the vegetative cells are going, like I said, are going to take up the safranin, and they're going to be red, and the spores should look green from the malachite green. All right, what do you know about Staph epidermidis? Is that a spore former? No. So how do you expect those cells to look? We know that they're cockeye, we know that they're round cells. Since they're not spore formers, they're all going to be red. Okay, so let me show you some examples of what we hope to see. This first picture, I just got this off the internet. I'm not even sure whether it's a Bacillus or a Clostridia or whatever, but I liked it because the colors are so distinct here. So here's a couple of free spores here. Definitely, I would call that green. Sometimes students say, oh, the spores are blue. No, I would say these are green. They should be green. The vegetative cells here are red. You can see a couple of cells here, like there's one down here where the spore is still within the vegetative cell. You can see it's kind of not right in the center for this cell. It's kind of more towards the end. It depends on what species of cell you have as to, here's another one, as to where that spore is located, whether it's central or terminal. Okay, so for instance, Bacillus Subtilis, they always have central endospores. So this was a student slide that one of us took a picture of. And again, so you've got the green spores, not quite as bright green here. The red vegetative cells, these are much shorter vegetative cells, almost look the same size as some of those spores. But some of these, then the endospore, some of them you can see the green within the vegetative cell, and that's pretty centrally located. So again, this is probably Bacillus subtilis there. What would it look like if we gram stained Bacillus subtilis? I don't think that we usually do that in class. But remember, the reason that we have a spore staining technique is so that we can see those spores better. If we're gram staining, are we using heat? Only to heat fix. That's just very brief. So we're not applying stain and heat together. So I suspect some of these smudges here, these are spores that you can just kind of barely make out. They've taken up a little bit of dye. You would probably miss that this organism was a spore former unless you could see a few cells with an unstained spot in the center of them. Again, the spore is not taking up the dye, so you see a little spot in there that's unstained. Okay, so purple rods, remember, these are gram positive. All right. And one other picture to show you, this is, I'm not sure which species of Clostridium this is. I believe this, I can't remember if this was a picture I took or not. It kind of looks like it might have been. It's a little bit harder to make out the colors here, right? We'll call these spores green, even though it's kind of, they're pretty light. But you can see the terminal spores. Here the spores kind of swelled up a little bit. Sometimes we call those tennis racket spores. But that's more often the Clostridium will have spores that look like that. So, all right, just kind of so you see a variety. Again, try to look at other people's stains as well as your own. We see a little bit of variation, but hopefully we'll get you know, lots of spores and lots of vegetative cells, both.